Let's begin today by stepping back and seeing where we are in our exploration of the concept of information in living systems. Remember that we've been looking for an information storing molecule that can do two things. First, it has to provide some sort of code for storing information about, we know, the structure of proteins. Second, it has to be able to be replicated in a fashion that preserves that information so it can be passed on to subsequent generations. So far in the course, we've shown how we came to know that DNA is the information storage molecule we're looking for. We've also seen how DNA can be replicated by a mechanism that accurately duplicates its structure and in so doing duplicates the information that it holds. As we saw last time, this replication mechanism depends on the complementary base pairing between nucleotides, as Watson and Crick first suggested. And it also requires the assistance of a whole number of proteins, the most notable of these being the enzyme DNA polymerase. The fact that DNA then can be accurately replicated and our understanding of how it is then satisfies our interest in the second of these two criteria for an information-bearing molecule. But what about this first criterion, the ability to store information? We still don't know how the code works. That is, we still don't know how the structure of a DNA molecule contains information about the structure of a protein. And that's where we want to begin to turn our attention to now. We do know something about the code. We know that the code only needs to specify a sequence of amino acids. That's because, as we've learned, the sequence of amino acids that comprise a protein more or less completely specifies that protein's structure and thus specifies its function. So to begin to understand how DNA works as a code, we first have to understand how the genetic code, or the genetic information flows in living systems in general. And that's the topic of today's lecture. Francis Crick, who we know is one of the co-discoverers of the DNA double helix, and has done a number of other very important things in molecular biology, proposed what he called the Central Dogma shortly after the DNA double helix was discovered. The Central Dogma is essentially a conceptual model for how genetic information flows in a cell. Now, there are two main points that are embodied in the Central Dogma that Crick proposed. The first point is the suggestion that information doesn't pass directly from DNA to proteins, but instead passes through an intermediate molecule. This intermediate molecule is the other main kind of nucleic acid that we've mentioned, ribonucleic acid, or RNA for short. The second point embodied in the Central Dogma is that information flows only in one direction, from DNA to RNA to proteins, and never back in the other direction. Crick put it this way, and I'm quoting him here: once information has passed into protein, it can't get out again. Well, let's follow the path of the flow of genetic information as is outlined by the Central Dogma. Information is stored in DNA, and as we might guess already, that information is stored in a sequential pattern of the bases along the DNA polymer. We're going to look at this in more detail a little later on. The information that's stored in DNA can pass to other DNA molecules when they are replicated. That is, the flow of information from one DNA molecule to another can occur in a circular fashion when that DNA molecule is copied exactly by the mechanisms that we've already discussed. This process we've termed replication. It could also be called DNA-directed DNA synthesis because it's a DNA molecule that's directing the pattern synthesis of more DNA. Now, the information in DNA can go elsewhere. It can also pass on to molecules of RNA. The process by which genetic information is transferred from DNA to RNA is called transcription. Transcription might also be called DNA-directed RNA synthesis. Note that the biochemical language of RNA, a nucleic acid, is the same as the biochemical language of DNA. It's just a specific sequence of nucleotides. Now, having the information that's stored in DNA transferred to RNA doesn't actually do us much good at this point. The function of RNA in this context is to act as an intermediate or a messenger, taking the information it's gotten from DNA and then passing it on to the process by which proteins are made. Now, we've heard about RNA before in a couple of contexts in this course. Now that it's starting to figure a little more prominently in our discussion, I'd like to give you some background on the chemical differences between DNA and RNA. The structure of RNA is pretty similar to the structure of DNA, with three important differences. The first difference is a subtle chemical one, and it's not really essential for our understanding of how RNA work, but it's what actually gives rise to the names of these two molecules, so I thought I'd mention it. The pentose sugar that's found in RNA, that sugar to which is bound a phosphate group on the one hand and a base on the other in nucleic acids, that pentose sugar is called ribose. Now, the technical detail here is that it has a hydroxyl group. Remember, that's an oxygen and a hydrogen bonded together, bonded themselves to the 2' carbon, the second carbon in the chain of that pentose sugar. The pentose sugar that's found in DNA has lost this hydroxyl group and in fact only has a hydrogen in that position. Now, I told you that's a subtle difference, but the names here are what I'm getting at. You see, because DNA is called deoxyribonucleic acid because the pentose sugar in DNA, having lost that oxygen, is called deoxyribose. So we have DNA and RNA, deoxyribonucleic acid and ribonucleic acid, respectively, based on the pentose sugar difference. Now, the second difference between DNA and RNA is that one of the nitrogenous bases of the 4 bases that are found in these nucleic acids differs. Instead of the base thymine that we find in DNA, RNA uses the base uracil. Uracil and thymine also are quite similar in their chemical composition, but they are different compounds. So what that means is if we look at a molecule of DNA, we'll find adenine, thymine, guanine, and cytosine as the bases along the chain. But if we look at a molecule of RNA, we will find instead adenine, uracil, guanine, and cytosine. So the uracil is essentially taking the place of the thymine. And in fact, in the complementary base pairing that goes on in RNA, the uracil again takes the place of thymine. So uracil will complementarily base pair with the adenine instead of thymine in an RNA molecule. Now, the third difference between RNA and DNA is in some ways the most significant. And that is RNA usually occurs only in single-stranded poly and not in any kind of double-stranded poly, like the double strand of DNA. A consequence of that is that RNA generally doesn't have any higher order structure. It only has the primary structure of the sequence of the nucleic acids that comprise it. Unlike DNA, for example, where we have a higher order structure, the double helix that emerges when these 2 strands of DNA come together. Now, as we'll see as we go along, there are, from a functional point of view, several different kinds of RNA. Then we're going to mention those as we come to them in turn. The RNA that we're talking about at this point in the context of the Central Dogma, we call messenger RNA for obvious reasons, or for short, we might call it mRNA. Okay, let's get back to the way information moves as is described in the Central Dogma. The information occurring in RNA can only go to one place in general, and that is to proteins. The information passes to proteins through a process that we call translation. Now, a key point to remember here is that the biochemical language changes at this point in the flow of information. It changes from the language, so to speak, of nucleic acids, which have their particular kind of biochemistry, to the language of proteins, polypeptides, which have a different kind of biochemistry. And that's going to become very important when we look at the details of how that information passes. A simple way to put this is that RNA and proteins don't talk together as easily as DNA and RNA can talk together because they're different kinds of molecules. Now, ultimately, it's the process of translation that takes the information originally stored in DNA, but stored there in a relatively useless state from the point of view of the functioning of the cell, because it doesn't do anything, to the relatively useful state which is the structure of a protein or of proteins, which are the things that perform all the critical functions of life. Okay, so the Central Dogma provides a framework for organizing our thinking about the molecular processes responsible for the synthesis of proteins ultimately directed by the genetic information found in DNA. Now, in this context, I'm prompted to note that we've been talking about molecular genetics for a couple of lectures now, and we've thrown the word gene around in this context a few times, but we haven't actually explicitly defined what we mean by the term gene. You can define genes in different ways in different contexts, or define the word gene in different ways in different contexts. In this context, at least as a first approximation, the Central Dogma makes clear that on the molecular level, a gene must be a segment of DNA. A segment of DNA that carries the code for making a particular protein. The Central Dogma also provides a framework for thinking about how the process for synthesizing protein from DNA might be controlled. You see, at each of these transitions, from DNA to RNA, from RNA to proteins, there are mechanisms at which we can control the flow of information, turning it on and off. We're going to look in detail at the mechanisms responsible for this gene regulation, as it's called, in the second section of the course when we look at development. Finally, and most importantly, the Central Dogma provides a molecular framework for understanding a key point about evolution. As I said, it says that information only goes in the direction from DNA to proteins, and never vice versa. This constraint has a profound effect on the way that genetic information in living systems can change over time, which is another way of saying that it has a profound effect on the way that living systems evolve over time. This constraint, to put it simply, is that changes in the genetic material of an organism can only occur through changes in the DNA itself. It can't be imposed on the DNA through other agents. And the only way that those changes can occur is through some sort of mutation, some sort of error or other kind of damage that's introduced into the DNA. Evolution is a change in the average characteristics of individuals and populations of organisms over time. And as we'll see later in this segment of the course, evolutionary changes must be somehow linked to changes in DNA. That much really should be obvious so far. So then the question is, how can we find, or how do we get changes in the DNA of an organism? The only way is if something happens to the DNA itself. There's no way for changes in RNA or proteins to somehow feed back and affect the structure of DNA. Think of it in this context. As you or I are living our lives, there are many things that happen to us that change us. Let's say, for example, we exercise. That kind of training will affect our shape, our size, how good our structure, our function is in certain ways. All of those kinds of changes ultimately are changes on the level of the proteins that are coded for by DNA. There's no way, however, for those kinds of changes to somehow feed back to our offspring. You can exercise all you want, but it won't affect the muscles of your offspring. Or conversely, if you have something happen to you, some sort of damage like a wound, you may have a scar that's permanent, but that scar will in no way affect the appearance of your offspring. We'll see later in the course, by the way, that the mutations that occur in DNA are random. The changes that occur in DNA are random and are not themselves influenced by the environment. And that'll become an important point in our understanding of how evolution by natural selection works. Okay, now, the word dogma, strictly speaking, means a doctrine that cannot be doubted by a true believer. That's quite a strong position, and one that's rarely, if ever, appropriately taken in science. Francis Crick used this term when he formulated his central dogma of molecular biology, but he later admitted that he didn't really know the strict definition of the word dogma. It's just that central dogma sounded better to him at the time than, say, central hypothesis or the central model or the big idea or something like that. So he called it the central dogma. Crick should have known better. For shortly after he introduced this term, researchers began to find some important exceptions to the central dogma. And it's these exceptions that I want to turn to now. These exceptions are relatively uncommon. And in that sense, they're truly exceptions that prove the rule. In general, the central dogma holds. What these exceptions do is illustrate more specifically the principles of how genetic information flows in living systems. As early as 1955, actually, just a couple of years after the discovery of the DNA double helix, and literally almost immediately after Crick introduced the term central dogma, Heinz Frankel-Conrat discovered an interesting virus. This is called the tobacco mosaic virus that appeared to lack DNA altogether. Now, we've talked about viruses already in the context of the experiment done by Hershey and Chase. You remember that's the experiment where they looked at where the different parts of the so-called T2 virus went when it infected a bacterial cell. And they were able to show that this virus was composed only of DNA on the inside surrounded by a central coating of protein. And they were further able to show that it was the DNA that was injected into the bacterial cell. And that's what caused the bacterial cell to change its activities, thus demonstrating that DNA was the genetic material. What Frankel-Conrat found in the tobacco mosaic virus was that actually it contained RNA instead of DNA. And we call these kinds of viruses RNA viruses. So in this virus, then, the flow of information is truncated compared to the flow that's described by the central dogma, going only from RNA to protein. When these viruses infect cells, it's RNA, not DNA, that is inserted into the cell they infect. And it's RNA that directs protein synthesis in the cell, and in that sense takes the cell over. DNA is simply left out of the information stream altogether. Since Frankel-Conrat's original description of the tobacco mosaic virus, hundreds of other types of RNA viruses have been identified. So this exception to the central dogma is actually somewhat common. This exception is also not really much of an exception, however, since the direction of the flow of information is the same as Crick originally proposed. It goes from nucleic acids to proteins. The trick that these viruses have to solve, however, is how to make more RNA without using DNA as a template for its synthesis. You see, because RNA normally occurs as a single-stranded molecule, not as a double helix, there's no inherent mechanism for complementary base pairing to provide a replication mechanism the way that complementary base pairing provides a replication mechanism for a molecule of DNA. So instead, these viruses had to come up with some other procedure to synthesize new RNA using only an existing RNA molecule as a template. And they do this in a two-step process. First, what they do is use the existing RNA template as a template for making a complementary strand. Now, they've synthesized a strand of RNA, but remember, because of the complementarity, the newly synthesized strand of RNA wouldn't make any sense because all of the bases are the opposite in order as the template from which it has been synthesized. So they then have to take this newly created RNA strand and synthesize yet another RNA from that. And in this two-step process, they create essentially a mold, which then provides a template to make a copy of the original RNA molecule. Now, another and even more startling exception to the central dogma was discovered in 1970 by David Baltimore, Howard Temin, and Satoshi Mizutani. What Baltimore and his colleagues found was an RNA-containing virus that was capable of synthesizing DNA, not RNA, from an RNA template. This discovery upset one of the key relationships embodied in the original formulation of the Central Dogma by showing that it's possible for information to flow in the reverse direction from RNA to DNA, completely at odds with what Crick had proposed. These kinds of viruses are now called retroviruses. Now, the trick that retroviruses have accomplished is to have an enzyme that directs the polymerization of DNA from an RNA template instead of a DNA template, an enzyme that we call reverse transcriptase. Again, remember I told you that the names of enzymes often make sense if you just sort of speak them out. This is a transcriptase. It's a transcription enzyme that goes in reverse transcriptase. When one of these retroviruses infects a cell, the reverse transcriptase reverse transcribes the virus's own genetic material, which is in the form of RNA, into DNA. And here's the really insidious thing. This newly synthesized DNA is actually inserted into the host cell's own chromosome. In other words, the DNA molecule of the host cell is literally broken apart and then has an additional chunk of DNA, which is from the retrovirus, inserted in, and then the whole molecule is stitched back together. So the DNA of the host is modified substantially by the activity of reverse transcriptase. Then, once this DNA has been inserted into the host's own genome, it will direct, using the host's mechanisms for transcription and translation, the production of RNAs and proteins, which are the RNAs and proteins that will go to build more retroviruses. Obviously, the discovery of retroviruses provided a profound exception to Crick's formulation of the Central Dogma. The discovery of retroviruses also was of great importance for practical reasons. This is because retroviruses are responsible for some of the most difficult diseases to understand and treat, including the HIV virus, which is responsible for AIDS. As I'm sure you know, the HIV virus attacks cells in the immune system, eventually rendering those cells non-functional and thus leading to the immune deficiencies associated with AIDS, which stands for Acquired Immune Deficiency Syndrome. The HIV virus and other retroviruses are particularly difficult to treat for two somewhat different reasons. First, because the HIV virus actually inserts DNA transcripts of its own genetic material directly into the DNA of the host organism, treatments for HIV have to be specifically targeted only to affect steps of the process that are unique to the virus's function. You see, you can't do something that more generically affects the ability of DNA to transcribe RNA or RNA to translate into proteins, because in so doing, you would turn off the cell's own functions completely, as well as whatever functions have been imposed on it by the added DNA. This is why the first treatments for HIV were designed to very specifically block the action of that enzyme reverse transcriptase itself, to prevent the reverse transcriptase from ever being able to add DNA to the host cell's chromosomes. An example of this approach, actually one of the earliest examples, and I'm sure you've heard of this, is the use of the compound AZT, or azithromycin. AZT is a chemical compound that resembles the base thymine, which is normally a base found in DNA. Because of this biochemical resemblance between AZT and thymine, reverse transcriptase often mistakenly identifies an AZT for a thymine, and when it is making a molecule of DNA, it will find an AZT that is base paired against an adenine, in this case, and it will add it to the growing string. Fortunately for the use of AZT as a treatment, AZT, because it's chemically modified as compared to thymine, actually doesn't allow the transcription process to continue. And the reason it doesn't is because AZT itself is not able to bind a subsequent nucleotide in the process. So that means that when an AZT molecule is added to the growing DNA polymer, the polymerization process just stops. So in this way, AZT shuts down polymerization of DNA specifically by the reverse transcriptase enzyme. And it's specific to the action, or I should say really relatively specific to the action of reverse transcriptase, because ordinary DNA polymerase doesn't make this mistake. When the cell is using its own DNA polymerase to make copies of its own DNA, it does not as often confuse AZT for a thymine. So DNA polymerase works just fine, whereas reverse transcriptase has a problem. At this point, there have been a number of similar kinds of antiretroviral compounds found, all used to block the action of reverse transcriptase. Now, there's a second reason that HIV/AIDS is difficult to treat that also has to do with the way the reverse transcriptase enzyme works. As compared to normal DNA polymerase, reverse transcriptase is kind of sloppy. It makes mistakes, and it makes mistakes much more often than mistakes are ever made by the normal process by which DNA is polymerized in replication. Now, a consequence of the fact that it makes mistakes is that there are genetic changes introduced into the information of the AIDS virus at a very high rate. And these changes, these mutations, are a substrate on which evolution can occur. Because there are so many more mutations of the HIV's own genetic material, what that means is that HIV can actually evolve very rapidly even within an organism. This is why HIV-positive patients often cease to respond to AZT treatment or any other kind of similarly acting retroviral treatment, because the HIV that's within them has mutated and evolved to avoid recognizing AZT, and that treatment no longer works. This has been a serious problem in developing treatments for patients who already have HIV, because if a treatment works at one point, at some point further on down the line, that treatment will stop to work, and this is why so many antiretroviral compounds have been looked for and found. Okay, let's get back to the central dogma. There is one key relationship in the central dogma that still seems secure, at least at present. And this is the statement that information can't flow from proteins back to nucleic acids. It can't flow from proteins back to RNA and then back to DNA. No one has yet found a case where a change in a protein can somehow induce or feed back a change in the information that's encoded in RNA or DNA. There is an interesting case, however. It's not really an exception to the central dogma because it doesn't affect this, or doesn't imply a backward flow of information, but it's an interesting case because it seems as though it's possible to change the structure of a protein, of one protein, based on information carried in another protein. This is the case of disease-causing agents called prions. These disease-causing agents are responsible for a class of degenerative brain diseases, which are called in general by the somewhat technical and maybe a little bit comical name, spongiform encephalopathies, which literally means sponge brain diseases. And they're called that because these diseases cause a degeneration of brain tissue in the individuals that are affected by them. Examples of these kinds of diseases are, for example, mad cow disease, which we hear a lot about in the news, or Kuru. Prions, the word prions is short for proteinaceous infective particles. They are a kind of protein. There is no DNA or RNA associated with them. Now, how could they work? For many years, researchers were looking for whatever infectious agent were responsible for these degenerative brain diseases like mad cow disease, and they were unable to find any. That's because they weren't looking in quite the right place. Then Stanley Prusiner recently, only a few years ago, identified a particular kind of protein, which he named a prion, that occurs in one shape in infected brain tissue and occurs in another shape in uninfected or normal brain tissue. Somehow, by a mechanism that isn't understood yet, Prusiner and others have suggested that these proteins interact such that the infected protein the disease-causing shape of the protein can interact with a normally shaped protein and cause it to change its shape. Now, as these proteins multiply, obviously the incorrect shape spreads through the affected individual. And this spread of a change of shape is a kind of transfer of genetic information. Well, prions push our understanding of how information might be defined and how it might be transmitted in cells. Suffice it to say now that what we want to do, now that we've looked at the big picture of the flow of genetic information in living systems, is turn our attention in the next lecture specifically to the nature of how information is encoded in nucleic acids.